Hello everyone. I just wanted to mention that I come across a lot of students who sort of struggle with, first of all, the purpose of studying Drosophila. Uh, they struggle with how you can actually use it for any sort of biology, understanding how the principles behind the organization or functioning of tissues. And so I decided that I'm going to put together a series that should be useful to students everywhere who have a basic understanding of genetics. And I will do my best to build bit by bit in such a way that you should be able to think about um, problems in biology and how you can use Drosophila to address them. So this is the first part where I will go through a bit about why study Drosophila. So Drosophila look very different from us. And so how is it possible that anything you can learn from Drosophila is relevant to human biology? That is a question that you might ask yourself. And it is important to remember that the way proteins and tissues function inside Drosophila or in mice is similar to how things work inside the human body. There are variations on the basic fundamental principles that produce this sort of variety and what looks like complete incompatibility. So again, just to reiterate, though humans look different from mice and flies, the basic principles of operation are actually very similar. Like humans have various organ systems. So here we're showing you the digestive tract, the nervous system, the principles that govern why your arm is not coming out of your hip, or tissues like the heart, your kidneys, have similar systems as well in Drosophila, right? So it is important to remember that the systems are similar and the governing principles, the genes and their regulation, are often similar as well. What those are, you cannot study in humans directly, and you should not. And you can do that in Drosophila. Now let's ask why Drosophila versus other systems. So Drosophila, first of all, let me highlight the fact that Drosophila are very similar molecularly. So to demonstrate this, what would be an experiment that would show this best? So if I had to show that two things are similar, if I could take one part out of one object and put in the same part or similar part from another object, and the first object starts working, that tells you that those parts at least are working similarly. So for example, a mutation in a gene called PAX6 in humans causes defects in eye development, right? And the same thing happens when you take away the gene that looks like PAX6 in terms of sequence, called iris, in flies. So the eyes of the Drosophila don't develop. Now, so what happens if you take the human gene and express it, activate it in all the parts of Drosophila? You can actually get eyes developing everywhere, right? Now, obviously, the first question is, can the fly see through all of these parts? No, because though the structure of the eye is there, the innervation and connection to the brain is missing, but the development of the eye can occur in many places outside where it is supposed to on a fly. If you express the human gene that is involved in eye development into a Drosophila, right? So this suggests that there is molecular similarity between human genes and Drosophila genes, right? So we're working with similar machines, but you should be able to control the behavior of a molecule inside a living animal at all times, right? And so Drosophila actually offer that kind of ease. So what you're seeing here is highlighted inside a living Drosophila. Its brain and central nervous system. You can target the function of specific molecules only in the nervous system if you want. You can do that if you want to target function of molecules only in the heart. You can do the similar thing if you want to affect only the flight muscles and pretty much a lot of other tissues as well. And within those tissues, you can target specific subsets. And so experiments can be performed in living animals, and that is important. Why is this important? Usually, if you want to change molecular function, take away a protein or hyperactivate a protein, you can technically, practically do it easily in cell lines, or in with just proteins in a test tube. But all of these experiments sort of decouple the function or the process that you are interested in from all the cues that it would receive inside a living animal. You will be able to understand the process, but it will not be regulated in the same way as in a cell line as it is inside a living animal, right? And so you have very clear control over molecular function at the cellular level in a living animal, which is the fruit fly, and that is hard to get in cell lines. So while you might discover new things that are happening in cells, they may never actually happen inside a living animal because they never reach that point, or endogenous control never allows for them to happen. And so it is crucial that we try and understand molecular processes inside a living animal. And so doing so in vivo is important. Also, if you want to do this in living animals, you can do this in other vertebrate models that we use, like mice. Except that you have to realize that taking care of mice is expensive. Taking care of flies is not expensive. Taking care of flies, starting with an adult male and a female, within 10 days you can get tens of progeny, right? And so you can actually start using all of these progeny for your experiments right away. Whereas with mice, it takes a while for them to grow. You have to care for them in that time, which means very regulated temperature, very regulated food over a longer period of time. So basically, you can use Drosophila for research in biology. So just to reiterate what I've said in very simple words, you can use Drosophila to understand the functioning of genes, transcripts, proteins, and behavior within specific tissues and cells because of the technical facilities it offers. What are these technical facilities? What are these tools? That is something that I want to cover during the rest of this series. Further, like I said, you can show that the proteins working in flies work similarly in humans and vice versa. This is not true for all proteins and genes, but that does not matter as long as your problem of interest can be addressed in Drosophila. And like I said, they're easy to use and relatively easy to care for compared to vertebrates. They result in large numbers, so you get, you are surer of the observations that you make because you can get really clear statistics. They have a short lifespan. They are quicker results. That is something that is great, especially if you're doing a PhD. You don't want your PhD to last 8 years. So you can get solid results quicker, and because these are fruit flies, the ethical concerns that apply to vertebrates like mice and rats do not apply here. So it's easier to get relevant information with greater surety and quicker. That's the important part. That's why Drosophila are a good model system. And what kinds of questions have already been used to have been addressed using Drosophila? You've got Nobel Prizes awarded for understanding heredity based on chromosomes, which use Drosophila. Mutagenesis and through X-rays received a Nobel Prize. Understanding, like I said, why your arm doesn't come out of your hip. Those principles were found in Drosophila. Again, a Nobel Prize winning finding. The protective mechanisms against pathogens won a Nobel Prize in 2011. Then circadian rhythms won a Nobel Prize in 2017. And these have been the starting point of understanding how vertebrate and human bodies function. Right? Again, Drosophila are great to understand the fundamental principles that govern the behavior of tissues inside a living animal. And from those principles, you can see how they are regulated and what variations exist in humans. Without these principles, you would not be able to do that in humans. If you did, it would be ridiculously expensive and you would not be as sure of your results. So, like I said, Drosophila are not human. I mean, I think we can all agree to that. But the development of circuitry in the brain, which governs how the brain functions, leading to behavior, and in cases where there are diseases, dysfunctions, all of that can be found out in flies inside a living animal. Drosophila don't get cancer, but you can understand how cells start proliferating. What regulates their proliferation inside a living system much better using Drosophila. The hereditary diseases can be directly tackled inside Drosophila just because it is so easy to grow them, and you have control over almost every nucleotide inside the animal. You can build the right genetic makeup that resembles a disease and see how much of it is responsible for diseases. And that's the best part. You can try to guess what intervention might correct this disease better than actually in any other model system. So in this series, I think you can expect to understand the theory behind using Drosophila genetics, meaning what is the makeup, how can you actually introduce new genes, how can you take away new genes, how can you knock down genes, take away their function. You can understand how you can hyperactivate a gene, a protein's function. You can also, once you understand those basic principles, how can you design these experiments yourself? How can you plan and write crosses, care for your flies? All of these details will be covered, and if you have specific questions, you can write to us at the d2lab@gmail.com. We are on X at the D2Lab and on Blue Sky at the d2lab.bluesky.social. Right? And so this is an end to the first session.